Hello friends, today we will learn about identification of bacteria. In this video, we will discuss about identification and characterization of bacterial strain, conventional methods, morphology of bacterial colony, growth in liquid media, staining, hanging drop preparation, biochemical test, antigenic structure, typing methods, pathogenicity test, antibiotic sensitivity test, and rapid methods we'll discuss in this video. So let's begin. After isolation of bacteria in pure culture from a specimen, it has to be identified. There are some necessary studies to characterize a bacterial strain. First is morphology of bacterial colony, growth in liquid media, staining, hanging drop preparation, biochemical test, antigenic structure, typing method, pathogenicity test, antibiotic sensitivity test. Now let's know about conventional methods. In conventional method, first is morphology of bacterial colony. The appearance of bacterial colony on culture medium is usually characteristic. That is, first point is its size in millimeters. Size is in millimeters. Example, pinhead size is characteristic of Staphylococcus, whereas pinpoint size is a feature of Streptococcus colonies. Second point is shape. It may be circular or irregular. Third point is surface, that may be smooth, rough, granular. Fourth point is elevation. Colony may be flat, rising, low, convex or umbonate. Fifth point is age, entire, crenated, lobate, undulate, ciliate. Sixth point is opacity. It may be translucent, transparent or opaque. Seventh point is color. Pigment may be produced by certain bacteria. Next point, eighth point, hemolysis. It may be produced by some bacteria, leads to hemolysis around the colony. Eighth point, consistency. It may be mucus, friable, firm, butyrous. Now let's know about growth in liquid medium. Commonly used liquid media are propellant, peptone, water and nutrient growth. Bacterial growth appears in many forms. Let's know about the forms. First is uniform turbidity, deposit at bottom, surface pellicle formation. 3 points. First is, let's know about uniform turbidity. That means most of the gram-negative bacteria grow in this form. Deposit at bottom. This occurs in growth of Streptococcus. Chains of this bacteria being heavier may settle down at deposit. Surface, third point is surface pellicle formation. All aerobes have tendency to grow on surface of media due to more content of oxygen present on the surface. Example, Pseudomonas species. Now come to staining. Staining methods are employed to examine smears prepared from the bacterial colony or liquid culture. Commonly used stains are first gram staining, Albert staining, gel staining, Gels-Nielsen staining, like this. Let's start with gram staining. Gram staining, it divides bacteria into gram positive and gram negative. It may be used for almost all bacteria. Now come to Albert staining. It is employed to identify Corynebacterium diphtheria, a causative agent of diphtheria. Gel-Nielsen staining. It differentiates acid-fast bacilli, that is AFB, from non-acid fast bacilli. This strain is commonly employed for identification of Mycobacterium tuberculosis and M. leprae, which are causative agents of tuberculosis and leprosy respectively. Both these bacteria are acid fast in nature. Next point is hanging drop preparation. It helps to distinguish motile bacteria from non-motile ones. Now come to biochemical test. Bacterial species differ in their capacity to break down different sugars. Some biochemical tests are based on presence of specific enzymes such as catalase, coagulase, oxidase, urease, gelatinase, etc. Now there are some other process biochemical test. Let's know about this test. First is sugar fermentation. Its principle. Let's know about its principle. To determine the ability of an organism to ferment a specific carbohydrate, that means sugar, incorporated in a medium producing acid or the acid with gas. Now come to procedure. Test organism is inoculated in a sugar medium and incubated at 37 degrees centigrade for 18 to 24 hours. Glucose, lactose, sucrose and mannitol are widely used sugars. Sugar media contain 1% sugar. Indicator used is Andread's indicator, a solution of acid fusin to which is added sodium hydroxide. Now let's know about interpretation. Positive, it may be positive, pinkish red, it is pinkish red, that means it's acidic. Negative, negative means yellow to colorless, that may be alkaline. Gas production can be seen as bubbles in Durham's tube. Now let's know about the examples of fermentation bacteria. Glucose fermenters, all members of Enterobacteriaceae, and glucose and lactose fermenters, that means Escherichia coli, Klebsiella species. Glucose and mannitol fermenter, that means Salmonella species. Indole production. Let's know about the indole production. Its principle. To determine the ability of an organism to decompose amino acid tryptophan into indole. Tryptophan is decomposed by an enzyme tryptophanase produced by certain bacteria. Procedure. Let's know about the procedure. Indole production is detected by inoculating the test bacterium into peptone water and incubating it at 37 degrees centigrade for 48 to 96 hours. 0.5 ml of Kovac reagent is added to the bacterial growth and gently shaken. Now let's know about Kovac reagent contents. It contains para-dimethyl amino benzaldehyde 10 gram, amyl or isoamyl alcohol, that is 150 ml, and concentrated HCl, that is 50 ml. Now let's know about interpretation. Indole positive, a red colored ring near the surface of the medium. Indole negative, that means yellow colored ring near the surface of the medium. Now let's know about positive and negative bacteria. Indole positive, that is E. coli, Escherichia coli, Proteus species other than P. mirabilis and Edward Silla. Indole negative, that is Klebsiella species, Proteus mirabilis. Now come to urease production. Its principle. To determine the ability of an organism to produce an enzyme urease which splices urea to ammonia. Ammonia makes the medium alkaline and thus phenol red indicator changes to pink or red in color. Now come to procedure. The test is done in Christensen's urease medium. The test organism is inoculated on the entire slope of medium and incubated at 37 degrees centigrade. It is examined after 4 hours and after overnight incubation. Christensen's urease medium contains peptone water, urea 20%, agar, phenol red. Interpretation: positive, that is pink color, negative, pale yellow color. Positive and negative bacteria: UDS positive, that is Klebsiella species, Proteus species, Yersinia enterocolitica, Helicobacteria pylori. And let's know about UDS negative, that is Escherichia coli, Providencia species, Yersinia pestis. Now come to citrate utilization test principle. It is the ability of an organism to utilize citrate as the sole source of carbon for its growth with resulting alkalinity. Now come to procedure. Solid to liquid media can be used. A bacterial colony is picked up by a straight wire and inoculated into either of these media. These inoculated media are incubated at 37 degrees centigrade for overnight. Simon's citrate medium contains agar citrate and bromothymol blue as an indicator. Original color of medium is green. Interpretation: Simon's citrate medium, that is positive, that is growth with an intense blue color on the slant. Blue color is due to the alkaline pH which results from the utilization of citrate. Bromothymol blue indicator is blue in alkaline condition. Negative: no growth with the change in color. Cosset's citrate medium: positive, that is turbidity due to growth of bacteria. Negative: no turbidity. Positive and negative bacteria: citrate positive, Klebsiella species, Salmonella species except S. typhi, and Citrobacter species and Enterobacter species. Citrate negative, that is Escherichia coli, Edward Silla, Salmonella typhi, like this. And now come to catalase test principle. Let's know about the principle. Certain bacteria have an enzyme catalase which acts on hydrogen peroxide to release oxygen. Procedure: let's know about the procedure. Pick up a few colonies of test bacteria with platinum loop from nutrient agar slope or plate and mix it in a drop of H2O2 on a clean glass slide. That should be 10 volumes. A positive catalase reaction produces gas bubbles immediately. Since blood contains catalase, culture on blood containing media may result in false positive reaction. Use of iron wire loop for picking up bacterial colonies may be another cause of false positive test. Positive test, that means, let's know about the interpretation. Positive test: immediate bubbling, easily observed, that means oxygen is formed. Negative test: no bubbling, no oxygen formed. Now come to positive and negative bacteria. Catalase positive: all members of Enterobacteriaceae except Shigella dysentery type 1, Staphylococcus, Micrococcus bacillus. Catalase negative: Shigella dysentery type 1, Streptococcus, Clostridium. Catalase test is primarily used to differentiate between genera Staphylococcus from Staphylococcus to Streptococcus. Sorry, it's correct. Correct is Staphylococcus to Streptococcus. Now come to oxidase test principle. To determine the presence of an enzyme cytochrome oxidase which catalyzes the oxidation of reduced cytochrome by molecular oxygen. Procedure: freshly prepared solution of 1% tetramethyl paraphenyl paraphenylene diamine dihydrochloride, that is oxidase reagent, is used. There are different methods to perform this test. A filter paper strip Soaked in the oxidized agent is smeared with test organism. In a positive oxidized reaction, the smeared area turns deep purple within 10 seconds. Oxidized positive control should always be included to find out the working of oxidized strip. Another method is to pour oxidized reagent on the surface of colonies. The colonies become purple within 10 to 30 minutes. This technique is useful to pick up Neisseria colonies from mixed growth on culture media. Purple colored colonies can be subcultured to get pure cultures. Now come to interpretation. Positive, that is deep purple within 10 seconds. Negative, no color change. Positive and negative bacteria, that is oxidase positive, Pseudomonas species, Vibrio species, Neisseria species, Aeromonas species, Alkali genes species, Plesiomonas species, Moraxilla species. Oxidase negative, all members of Enterobacteriaceae and Stenotrophmonas maltophilia. Now come to phenylalanine deaminase test. To determine the ability of an organism to deaminate phenylalanine to phenyl pyruvic acid, in short PPA, this test is also commonly called as PPA test. Now come to procedure. Let's know about the procedure. A medium containing phenylalanine is inoculated with a growth of bacterial culture and incubated at 37 degrees centigrade for overnight. A few drops of 10% ferric acid solution is added. If PPA is formed, green color is produced due to reaction of ferric chloride with PPA. Now come to interpretation. Positive, green color, negative, no color change. Now let's know about the positive and negative bacteria. Positive bacteria, that means PPA positive, is Proteus species, Morganella species, Providencia species. Now come to PPA negative. All members of Enterobacteriaceae are the negative, PPA negative. Now come to methyl red test. Principle: this test detects the production of sufficient acid during fermentation of glucose by bacteria and sustained maintenance of a pH below 4.5. Now come to procedure. The test organism is inoculated in glucose phosphate broth and incubated at 37 degrees centigrade for 2 to 5 days. Then add 5 drops of 0.04% solution of methyl red. Mix well and read the results immediately. Interpretation: positive, if positive, that means red color, negative, yellow color. Positive and negative bacteria: MR species, Escherichia coli, Yersinia species, Listeria monocytogenes. And now come to MR negative: Klebsiella species, Enterobacter species, Vogus Proskauer VP, that is VP test or acetoin production test. Now come to principle. This test depends on the production acetyl-methyl carbinol, in short acetoin, from pyruvic acid in the media. In this presence of alkali and atmospheric oxygen, acetoin is oxidized to diacetyl, which reacts with alpha naphthol to give red color. Now come to procedure. Test organism is inoculated in glucose phosphate broth and incubated at 37 degrees centigrade for 48 hours. Then add 1 ml of 40% KOH and 3 ml of a 5% solution of alpha naphthol in absolute alcohol. Now come to interpretation. Positive, that means pink color within 2 to 5 minutes depending on deepening to crimson color in 30 minutes. Now come to negative, colorless for 30 minutes. Now come to positive and negative bacteria. BP positive means example Klebsiella species, Enterobacteria species, Eltor vibrio, Staphylococcus. BP negative, Escherichia coli, Micrococcus. Now come to indole. Indole, MR, BP and citrate test are very useful in the identification of enteric gram-negative bacteria. These tests are commonly referred to by the Singla IMVIC test. Now come to nitrate reduction test. Principle. In this test depends upon presence of an enzyme nitrate reductase, which reduces nitrate to nitrite or free nitrogen gas. Now come to procedure. The test organism is grown for 5 days at 37 degrees centigrade in a broth containing 1% KNO3. Then 0.1 ml of the reagent is added. That means sulfonilic acid and alpha naphthalamine is added. Now come to interpretation. Positive, if positive, then red color develops within few minutes, and if negative, then no color development. In case of no color development, it indicates either nitrate has not been reduced or nitrate has been reduced beyond nitrite to nitrogen gas, which reagents will not be able to detect. To interpret this, zinc dust is added to the test. Zinc reduces nitrate to nitrite, so development of a red color after addition of zinc dust means that the organism was unable to reduce the nitrate to nitrite, that is negative test. However, no color development after addition of zinc dust indicates positive test, as nitrate has been reduced to nitrite and further nitrogen gas. Now come to positive and negative bacteria. NO3 reduction positive. All members of Enterobacteriaceae, Branhamella, Catarrhalis. NO3 reduction negative, that means Haemophilus ducreale. Now come to triple sugar iron test, in short TSI test. To determine the ability of an organism to attack specific carbohydrates incorporated in a growth medium with or without the production of gas, along with the determination of possible hydrogen sulfide production. Now come to procedure. TSI is a composite medium which contains 3 carbohydrates, namely glucose, lactose and sucrose, and also ferric salts for testing H2S production. The concentration of lactose and sucrose is 10 minutes that of glucose in the medium. Phenol red is incorporated as an indicator. This medium is widely used and is in the form of the butt and slant in the test tube. Medium is inoculated with bacterial culture by a straight wire pierced deep in the butt. It is incubated at 37 degrees centigrade for overnight. Bacterium attacks simpler sugar, that is glucose, first, and then the lactose and the sucrose. The test is read after 18 to 24 hours and not before 18 hours. If the test is read before 18 hours, the glucose fermentation produces sufficient acid to make both slant and butt. Yellow means acidic, and thus giving a wrong interpretation of A by A. Refer interpretation. After 18 to 24 hours, the glucose concentration is depleted in the slant and butt. The organism begin oxidative degradation of the peptone present in the slant, resulting in alkaline products which change the indicator to a red color. Fermentation of glucose in the bud produces a larger amount of acid, overcoming the alkaline effect of peptone degradation. Therefore, the bud remains yellow. Certain bacteria produce H2S, which is a colorless gas. H2S combines with ferric ions to form ferrous sulfide as black precipitate and thus blackening of medium. Interpretation: yellow color fermentation of carbohydrate and red color means alkaline, that is no fermentation. Bubble in the bot, that means gas is also produced during fermentation of carbohydrate. Now come to blackening of the medium. H2S production. Blackening means H2S production. Various possible combination of the different TSI reaction are now we will discuss. K by A, that is red or yellow, glucose fermentation. A by A, that is yellow by yellow, glucose fermented. Lactose and sucrose fermented. K by K, that is red by red, neither glucose, lactose nor sucrose fermented. K means alkaline, A is acidic. Now come to hydrogen sulfide production. Principle: to determine whether hydrogen sulfide has been liberated by enzymatic action from sulfur containing amino acids, producing a visible black color reaction. Now come to procedure. Organisms are cultured in media containing lead acetate for ferric ammonium citrate or ferrous acetate. They turn them black or brown. Another method is to grow the organism in culture tubes, inserting a filter paper strip impregnated with lead acetate between the cotton plug and the tube. Blackening of the paper indicates H2S production. Interpretation is positive means black color, negative means no color change. Now come to positive and negative bacteria. H2S positive means Proteus mirabilis, Proteus vulgaris, Salmonella species with some exceptions. H2S negative, that bacteria are Morganella species, Salmonella paratifi A, S. cholerae, Swiss. Now come to growth in casein. Principle is to determine the ability of an organism to grow in a medium containing potassium cyanide. Procedure: inoculate peptone water containing 1 in 13,000 concentration of potassium cyanide with test organism. Incubate at 37 degrees centigrade for 24 to 48 hours. Interpretation: positive means turbidity due to growth, negative means clear, no growth. And positive and negative bacteria. Positive casein test means Klebsiella species, Citrobacter frondi, Pseudomonas aeruginosa. Negative casein test bacteria are Salmonella species, Escherichia coli, Alcaligenes faecalis. Now come to antigenicity test. Antigenic structure. Biochemically identified organisms are further confirmed by agglutination or precipitation reaction. Unknown bacterial antigen is identified by non-specific antisera by slide agglutination test. Salmonella typhi or other Salmonella can be confirmed by agglutination with the specific antisera. Now come to typing methods. This is useful for epidemiological studies. All this test can identify up to species level through intraspecific differentiation is not possible. Hence, typing methods like bacteriophage typing, bacteriocin typing and others are very useful for intraspecies differentiation. Typing methods help to find out the source of infection in epidemics or sometimes in outbreak of food poisoning. Now come to pathogenicity test. Guinea pigs, rabbits and mice are mostly used for pathogenicity test. These animals may be injected by subcutaneous, intramuscular, intraperitoneal, intravenous or intracerebral routes depending upon the organisms to be tested. The identification of organism is carried out on the basis of post-mortem findings and cultural characteristics. Now come to antibiotics test sensitivity test. The isolated bacterium is subjected to antibiotic sensitivity test in vitro for selecting appropriate antibiotic for therapeutic use. Different techniques for antibiotic sensitivity test now we will discuss. Now we'll discuss in next video, in separate video. Okay, now let's know about rapid method. Conventional methods take days to identify and characterize the isolates, while rapid automated methods take only hours. Detection of specific enzymes, toxins, antigens or metabolic end products of the isolates made the identification simpler and rapid. For example, gas liquid chromatography, in short GLC, is based on the identification of specific fatty acids produced by the organisms. This technique is very useful in identifying obligate errors. Polymerase chain reaction and nucleic acid probes carrying specific DNA and or RNA based sequences are some molecular methods being widely used for identifying microbes. Here in this chapter I have completed this chapter. Any questions regarding this you can ask me in the comment box. There are many short notes like indole production, urease test, citrate utilization test, site catalase test, oxidase test, PPA test, MR test, BP test, TSI nitrate reduction test. These are all questions in this chapter. So you have any questions you can ask me in the comment box and subscribe my channel for to know more updates. Thank you.